Oh, true. I need to disconnect the sound here, otherwise it'll get crazy.
So we're going live. So hello, everyone. Thank you for joining us today in our Open Neuroscience series. We have Loren Looger. Loren is a researcher at Janelia campus. He uses protein engineering to create tools for studying and manipulating the brain and other biological samples. His team combines computational and evolutionary methods to create new reagents for use in characterizing and manipulating the assembly and functions of neural circuits. The team designs and tests these reagents and other tools, collaborating with other scientists. As you may already know, we all use their tools. So their projects include creating sensors for calcium, neurotransmitter, neuromodulators, optogenetic effectors, and engineering viral capsids and probes for EM. And recently, they have designed proteins and pathways for carbon capture and other environmental remediation. Maybe that's something that we can discuss. And ultimately, the team's goal is to be able to enable better science in diverse fields across the globe through collaboration and innovation. And I must say that this lab has always been very on the front of sharing everything, being super open about what they're doing, being super open to collaboration and to basically sharing the knowledge amongst everyone and improving the tools that we all use. So I'm more than happy to host Loren Looger here in our series. So without further delay, I give you Dr. Loren Looger.
All right. Thank you for having me, guys. I really love what you guys do. So I was super excited when I found a request to volunteer. So I made this talk a bit different for this audience. It's, I'll cover our latest stuff, but I also made it more of a big picture talk to try to, if you have a tough problem in the lab and you're trying to figure out, oh, do I use this reagent or what Gren lens do I use, etc. I tried to make an overview of here are the various places you go to get your questions answered. And I also give shout outs to a lot of people. So I'm going to present a lot of work that's not mine, but these are great people and they're super collaborative as well. So yeah, I do this anyway, but in the spirit of open neuroscience, I have my slides will be available in addition to the recording, and you can reuse them as you see fit. Everything I'm going to present is available in many different formats, and I'll give you specific links to how to find that. And I mean, this is tougher in a Zoom audience, but in live audiences, I hope that people would interrupt and ask questions. And, as Matthias said, we're super collaborative, always looking for new collaborators. And so if you have an idea, please email me or call me or whatever. These are the things that I will briefly overview. So links to try to get you the answers that you need for your problems, and also find out like, oh, I didn't know that thing was available. And then these are the topics from the lab that have recent developments that I will try to, again, I'm not gonna try to overwhelm you with data. Just email me and I'll send you, data, preprints, whatever. Again, we're just trying to want to get on your radar screen as to what's available. So everybody needs fluorescent proteins. So I think by far the best resource out there for figuring out what fluorescent proteins there are, their spectral properties, their sequence, how they were engineered, photostability. This is a measure of how well it does as a fusion, so how monomeric it is. is definitely FP-based by Tally Lambert. So fpbase.org. You can find all the fluorescent proteins. They have the sensors as well, except that, you know, I see I've been a naughty boy and we haven't uploaded the photostability for our GCamp variant. So I'm going to do that this weekend, get that uploaded. So, you know, everybody needs DNA. So if you want to figure out what plasmids are available and their sequence and how to get them, obviously the resource you go to is Adgene. So just adgene.org. They have a nice little search interface. You type in what you're looking for, and they've always had plasmids. And within, I'd say, the last 2 years or so, they've started to offer live AAV preps in collaboration with the UPenn Viral Core. And so these are, we get these all the time, super high quality. So I highly recommend them. And, you know, so this just shows, you know, this is unpublished. I'm going to show you some brief data about this, the GCamp 8 series. You can type in and it tells you, you know, hey, you can get the DNA, or right now they have AAV1 and AAV9 that they're shipping as live virus. If there's anything on Adgene that, if you want like a different AAV serotype or anything like that, I find that they are super responsive by email and even their Twitter. I didn't have time to find a thread, but there are a lot of threads of like going back and forth about saying like, hey, we want AAV5. So definitely interact with them, and they're really responsive in terms of making what you need. Something else that they did, so around the same time that they started offering the live AAV preps, so you don't have to make your own AAV virus all the time, they started this really cool open science initiative looking at the expression patterns of different AAV serotypes. So I forgot to put the link right here, but if you just type in Adgene AAV data hub, it takes you right here. And so then you get this, you know, encyclopedia of entries. And so you can see, you know, what was the species, what was the brain region, what was the serotype, promoter, the payload, and then you click on each one and it takes you to, in most cases, just a single image, but I think they're trying to populate with more image stacks so you can scroll through and even get an idea of what cell types are expressed. And this is the brainchild of Melina Fan at Adgene, and she's also super responsive. So we need to deposit more data here, but it's a really good resource to have. So while we're on the topic of AAV, I just wanted to give a shout out to some of the best cores out there for getting things produced. I already mentioned Adgene, UPenn. A relatively new player in the game is the Canadian Neurotransmitter Sensor, but they have a rapidly growing catalog and their preps have been really high quality. So we always send things to Adgene because they do the plasmids as well. And we're starting to get better about sending things to alternative cores as well. And other good ones, just give shout outs to Salk, Minnesota Viral Vectors, UNC, and then Janelia has a viral core too. So that is brainchild of Kim Ratola. She's moving to UNC to start her own lab, handing it off to Hyuna Yi. Right now they do, this is, you have to be a Hughes investigator to get the easy way to the preps. There are exceptions, so they can make AAV, rabies, lentivirus. They're also really good about making the protocols open source. So Chem and Huna have really optimized the AAV production protocols a lot. And so that's right on the website too. So a lot of people use Cre-dependent payloads, and I wanted to bring something to your attention. You may be having problems that you didn't know you had. And so I want to alert you to the problems and offer some tips to solutions around them. So, so this is, this article that I'm taking these images from deals with Cre-dependent payloads in an AAV vector backbone, but you can get issues in other formats as well. And so maybe you've seen this before. So here is an AAV with a Cre-dependent payload injected into a mouse that should express in this one should express in somatostatin positive cells. This one should express in parvalbumin positive cells. And the green and the red should overlay, but what you see is that you are expressing in some cells that you shouldn't be. So that's a big problem, particularly if you're doing something like optogenetics or another effector, because if you're doing imaging or labeling, you could just maybe ignore those cells. But if you're firing your effector in cells you didn't intend to, then that's a real problem. And so what's going on is not terribly surprising, is that the lock sites are supposed to recombine in the presence of Cre, but depending on the specific protocol that people used, there can be a little Cre-independent recombination. And so then your tube of stuff that is supposed to be Cre-dependent is about 5% Cre-independent. And so you get this backbone. And you can see this by running it out on a gel, is you get stuff that is pre-recombined before it's gone into your animal. So there are various ways to improve this. This is discussed in those AAV protocol that I linked to earlier. The main thing is to make sure you don't overgrow your cells, either in bacteria or in the mammalian cells that you're using to produce the virions. And if you're super careful, you can also gel purify the stuff that has recombined. So just keep this in mind if you're doing something with a Cre-dependent payload and you're getting some weird results. This may be the explanation. So I told you about AAV resources. There are also good resources out there for lentivirus production. One that I would love to give a shout out to, they do a good job with open source lenti protocols, as well as having all the latest optogenetics reagents available, is the Stanford Optogenetics Core. I forgot to put the address on there, but it's easily findable. They do a really good job. So a lot of people will not benefit from transgenic animals in their work. So this has been, fortunately, this has been growing a lot in recent years, of these becoming easier to find and obtain. So obviously, mouse is the model organism of choice for many people. Pretty much everyone knows that you go to Jackson Labs to get your mouse line. They have a nice little search interface, so you can find all the latest lines. There are some that are much less well known. Like I didn't even realize that this was online until I did a Google search this morning, is that the transgenic rat. Center is up and running. And so, you know, you can get your Taiwan GCamp6F transgenic rats. Bloomington Stock Center has been around for a long time, and they are really good at getting the strains and the plasmids out to everyone. For fish, it's a little less well developed. ZFin does a great job of collating the information on lines, but for various reasons, it's hard to house thousands of transgenic fish lines and keep them maintained. So you typically have to get import either the animals or frozen sperm from the researchers. But ZFin is a good place to go to find out what's available and whom you need to email. That used to be the case with C. elegans, and for years I just said, you know, email my collaborators. They're good at sending the worm lines. The University of Minnesota, in addition to having the great AAV core that I already told you about, has a good C. elegans stock center too. And so you can get animals shipped for a lot of those strains. And, you know, there are other further afield animals, of course, but, you know, like there's lots of transgenic marmosets, but no core available for those yet. Wanted to give a shout out to my friends and collaborators at UCLA producing open source microscopes. I think they would be a great speaker in this series if you invite the UCLA Miniscope team. So they have everything on their website, miniscope.org, plans, part numbers, so you can build a fully functional Gren lens-based Miniscope for, you know, probably, I think it's around $15,000 now. So that's pretty amazing. And they offer you the whole package. So software, hardware, firmware, etc. So I really recommend you guys to invite them and have them show you the latest, because they've been really pushing the envelope in terms of always adding new functionalities like multi-site imaging, simultaneous optogenetics, and functional imaging. This is a pretty rapidly evolving space. In the same vein, I wanted to give a shout out to there's a number of people developing open source software for things like cell segmentation and analysis of functional imaging data sets. The two people that I really want to give credit to are both at Genelia, Marius Pachtaru and Carson Stringer. Although, you know, there's a pleasing number of labs developing open source software in this realm, certainly more than that, you know, even 5 years ago. So this is moving pretty fast as well, but they have some stuff, you know, obviously all their code is available at GitHub and downloadable from Genelia, but they've made some really cool web server interfaces to kick the tires. So this is CellPose, an algorithm for segmentation. So you can just drag your JPEG of the image you want to segment, or they show that they can segment, you know, fluorescence, DIC, EM, etc. And it, you know, a couple seconds later, you get the segmented image out. So that's pretty cool. For functional imaging data sets, they have something called Suite 2P. you know, optimized for 2-photon functional imaging data sets, but I imagine it would be pretty adaptable to things like Miniscope, like one photon methods as well. So this, I think, is a bit too unwieldy to have as the web server, but you can download, and they have like 3 or 4 tutorials on there. So it does pretty much everything for you. You know, steeper learning curve than CellPose, but pretty impressive in terms of segmenting your cell bodies, you know, from your regions of interest, trying to, you know, background, subtract, pull out the traces from each ROI, and you know, so it's pretty cool. It can do this pretty much in real time. You start whipping up your the traces for each cell body or other ROI, and it can do this for an impressive number of sites. So this is a data set generated from microscope made largely by Carl Svoboda and Dan Flickinger at Genelia, that does this serial section 2-photon imaging of, you know, like this is most, I believe this is most of sensory and motor cortex here. Pretty impressive. And so Suite 2P can handle all these cell bodies and generate thousands of traces at the click of a button. I know less about this, but while I'm giving a shout out, they have spike sorting on there as well with Kilosort, and mouse body and facial position tracking with FaceMap. A couple last things I want to actually, yeah, I think this is the last thing I want to tell you before I tell you about our stuff, is a nice community that has developed in this area, and I encourage you all to join and ask your questions and provide your answers, is the Neuromethods Slack. I find this super interactive. I have usually a couple threads a day going. You know, I spend a lot of time in the GCamp channel. Virus gets a lot of action, you know, opto, mouse lines, etc. So join. Let's grow this community, keep it lively, and bring more and more expertise here. So does anybody want to jump in with any questions before I start talking about what the lab has been up to? Okay, so I'll just plow ahead, but please, I know it's hard in Zoom, but I you know, please, you know, raise your hand, butt in, whatever. Yeah, so I figure if people want updates on any one thing, they probably want updates on the calcium indicators. So, you know, the lab, we've been developing GCamp for 14 years. People were developing GCamp for 5 years before we came along. You know, this is a very mature scaffold at this point, but it's, you know, I've been saying for almost a decade that I thought GCamp was almost done, that we, you know, we just couldn't make it better. We were getting diminishing returns, but time and time again, I've been proved wrong. And this is it's the gift that keeps on giving, and I'm really just blown away. So most everything I'm going to present is a lovely collaboration with the Genie project at Genelia, and this is Jan's work that I'm going to show you here, the development of what we call the JGCamp 8 sensors. So what we did with these sensors is we messed with a part of the protein structure that we hadn't much before, which is the interface of the peptide. We used to call it the M13 peptide, but you know, several people corrected us. This is properly called the RS20 peptide. But anyway, the calcium, the calmodulin and the calmodulin binding peptide, we really screened like 50 or 60 peptides here, and then re-optimized the linkers. Took about 5 years, but what we have are the biggest win of these sensors is that they have much faster kinetics, both rise time and decay time. And the beauty of this is that when you have a faster rise time in particular, you get a greater signal and noise. You get a bigger fluorescence response because although the calcium trace is, you know, much slower than the action potential itself, it is still pretty fast. And so when your sensor is slow, then by the time your sensor starts to build up, the calcium trace is already going down, and so you miss a lot of it. So with the new sensors, so, you know, kinetics only really makes sense if you look at like the single spike response, and you can see they're much faster than the previous best in class, GCamp 7F, much faster than GCamp 6S, and much greater signal and noise. And if you can zoom in to the rise time to the single spike, you see, you know, you're getting like a 10 millisecond, maybe even 5 millisecond rise time from single spike. So really many-fold faster than the previous generation. So they are, you know, still great out to 10, 40 action potentials or so. One bad thing about the sensors is, I mean, you know, there's no perfect sensor for every application. You want to have tools that solve most people's problems, but certainly from generation 6 on, we've started to make the multiple flavors. So we'll make like S is sensitive, F is fast, M is medium. A couple times we've had other flavors as well. So you can see that with the GCamp 8 sensors, by the time you get to these really long spike trains, you're losing a bit of the dynamic range. So if your question is let's say you have like an invertebrate sensory neuron that has this huge range in spike frequencies, and you really need to sample a large dynamic range of that, you might want to stick with an older generation sensor like 6S that could, you know, maybe better tell you, you know, the difference between 1 hertz and 400 hertz spiking. But if you're interested in, let's say, single events or spike timing, then you definitely want the new generation of indicators. So that was in cultured neurons. This is in vivo. Martin Rocha and Carl Svoboda acquired this data set. So this is 2-photon imaging with simultaneous cell-attached electrophysiology. So you can see everything looks really nice. The numbers are for a certain number of spikes in a short little train, and then the asterisk represents singles. And so you can see you're getting pretty robust responses to singles, and that the temporal resolution of spike timing is really quite good, particularly for the 8 fast, so F, but even 8 sensitive is a fair bit faster than the previous generations. And just to quantify that data, so again, you can see that the really fast rise times and the somewhat faster decay times held up in vivo. And this is the quantification across a lot of cells, a lot of animals. You can see that, you know, 8-fast has this like 8 or 10 millisecond rise time, and much faster decay time as well, while still having better amplitude, as I mentioned before. So that was Cortex. Our collaborators, Sam Wang and Joey Broussard at Princeton, did it in another in vivo prep. I'm not going to show you the data, but it worked well in Purkinje neurons in the cerebellum, which was really cool because the aficionados in the audience will remember that Purkinje cells have really interesting simple and complex spikes. And you really need the power of a fast indicator to tweeze apart the exact mechanics of what's going on. So just to wrap up that section, if you really have a need for speed, then you definitely want 8-fast, which 4-fold faster rise time than best-in-class 7F, bit, you know, much faster, but a bit less faster decay time. Sensitive is twice as sensitive as previous best-in-class, but still twice as fast as previous best-in-class. And then M sort of, you know, averages, it's still pretty fast and still pretty great sensitivity. So those are the 3 main flavors we're offering. Everything's available. So all the plasmids and AAV are shipping from Adgene already. Misha Arens' lab has characterized the transgenic fish. That looks good. Flies, this is taking a bit longer. The data so far suggests that the signal change and kinetics hold up but just fine. But currently we get a little bit of a hit, about a 2-fold hit in baseline fluorescence that makes them a bit hard to see. Because, you know, the fly people know that all the activity is in the processes and not in the cell body. And so, I mean, you can express a like a red reference protein for, you know, focusing on your region of interest. But it's much easier if we can just get this, if we could double the baseline fluorescence in flies, while keeping these properties the same, then I think that'll be much better. And so we're trying that right now. We think that the expression level just took a bit of a hit. So we're trying some protein stabilization tricks. Mice, the Genie project is making a lot of mice are already in the works. There's a Thy1 mouse for middle of the road 8M. Should be characterized by the end of the year. There is a TET reporter mouse with 8S, same time frame. And then there are improved Tiger 2 locus Cree-dependent mice for both 8M and 8S. But for various reasons, this locus takes a bit longer. So we hope that those are available at JAX by, you know, maybe summer of next year. And then just a couple last little details. We don't see any greater cytomorbidity from overexpression of these sensors. You know, the rate of cells getting G-camped out and nuclear filled looks the same. It doesn't look like it's any slower to diffuse in tissue. We are making a quite a diversity of fusions and promoter combinations. And a couple of these are at Addgene already. I think the H2B is coming online first. So if you fuse to H2B, that gives you much better segmentation of many cell bodies at the same time. And each of these other fusions offers its own little advantage. Quickly want to tell you about the other two major colors of Geckis. So for various reasons, yellow sensors have a lot of appeal. This is primarily driven by the development of SLAP microscopy by Kasper Podgorski at Genelia. That, so I'll just show you that on the next slide, lets you get really fast frame rate, 2-photon frame rate imaging of really large fields of view. So this is like almost 2 kilohertz imaging. This is not, so I'm showing you data from a yellow eye glue sniffer, not YCamp, but it's similar. And so glue sniffer is fast, as I'll show you in a couple minutes. Calcium is a bit slower. So you, instead of having the super fast speed, you can trade that for volumetric scanning, which might be more useful for YCamp. And reds, of course, are the other super useful color. So again, Genie, these are coming online pretty soon. We'll have Tiger 1 mice for JR Gecko 1A and also Rosa 26 lock stop locks. So hopefully by the end of the year, these mice will be available, and we're pretty overwhelmed with stuff. So we're actually actively looking for collaborators to help with functional imaging of these mice. So if you're interested, just drop me an email. And we're trying to make 2.0 versions of the JarCamp and JarGecko, but that's actually, it's really hard. It's by far the hardest color. So hopefully better news next year. So this is not my work. Wanted to give a shout out to a great team at Genelia, Eric Schreider and Luke Levis, who are still here, and Ahmed Abdelfatah and Claire Deo, who have since gone on to start their own labs. And then again, big help from Genie. So these are the currently best in class voltage indicators. So the oldest one that's been out, I think like 3 years, is Voltron, that uses the archaeodopsin voltage sensitive fluorescence of the retinal in the membrane. And instead of fretting with a fluorescent protein, as was previously done, they fret with a halotag covalent capture enzyme, and then a really bright and photostable Genelia fluor dye. And so that gives you much greater brightness and photostability, with, of course, the pain that you have to add a small molecule component. But for some things, this is really easy. You know, flies, you can put it in the food. Fish, you can put it in the water. Mouse, you can inject it in the tail vein. But I think it's a small price to pay for the huge advantages that you get in signal and noise and photostability. So the first one that was published was Voltron. where just like the earlier based earlier archaeodopsin based sensors, you get a negative deflection upon fluorescence increase. They since found mutations that inverts that signal. So this is called positron. I think that was published last year. They've got a Voltron 2.0 that I think is coming out hopefully by the end of the year. And then, okay, I think okay, my slides froze. Crap.
Matthias?
It appears that we're having some issue with the slides. We'll be back soon once we figure out about the frozen problem. By the way, you can join the Zoom meeting by clicking on the link on the YouTube description. We'll be back with Loren soon when he solves the technical issue that he's having, and we'll be back live then.
Hey everyone, I'm super sorry. Okay, where was it?
Hi, welcome back.
Yeah, so I'm so sorry.
It's okay. It's good. I think we actually can use the interruption for some questions. Here are some people asking about the red indicator, red calcium indicator for 2P spine imaging, where you have idea whether there's something good for, yeah, some red indicator for 2P spine imaging.
Yeah, so actually, yeah, I'll talk about that a bit on the next slide.
Awesome.
I mean, JR Gecko 1A does work pretty well under 2-photon. I mean, the reds are obviously way more complicated than the greens. You get things like you know, they blink, they you know, they're much less photostable than the greens. But I would say that JR Gecko 1A definitely best in class for 2P spine imaging with the fully genetically encoded indicators. But on the next slide, I'll give you a hint of something that might happen in the future to make the situation better. Any other questions now?
I think there was some other question, but you actually answered it with your presentation, so we can move on.
Okay. So that was Voltron. So that used the opsin-based voltage detection. That was always fundamentally limited by the fret with the retinal. Fret takes a big hit out of your signal and noise, and the retinal is a very poor, very dim fluorophore. So the next generation tools that they've made, they go back to using the voltage-gated ion channel-like voltage sensitive domains, the voltage paddles, usually from the voltage sensitive phosphatases, and then replacing the FP with the halo tag and the dye. So, you know, where it was in this extracellular loop, the genetically encoded one was called ASAP. And so since it's with the halo tag, they called this version HASAP. That one's really fast. Again, super bright and photostable. Marius and Carl did, believe, 15 minutes straight imaging of Voltron. I'm not sure if they got into HASAP yet, but compared to most of the fully genetically encoded voltage indicators, can usually bleach away in a couple seconds. So much more photostable and different topology. Using the arc light topology, you get arc light, and again, that works pretty well too. What I was saying about the potential for red and far red calcium indicators is that they've used the same trick of replacing a fluorescent protein with a halo tag and a dye to make something they called halo camp. The signal and noise of these things is not as good as the G-camps, obviously, and I think the kinetics are, you know, less. I mean, it's a new scaffold, so of course it can't be as good as something that's been improved for almost 20 years. But the really, I think the long-term potential of this is the ability to use far red or even infrared dyes with this. The downfall of this technique is that the dyes are kind of greasy and they don't always load very well. And particularly as you make them redder, they get bigger and longer and can be more hydrophobic and sticky. But I think that there's a chance that maybe within 5 years, they have a combination of halocamp and a dye where you're, maybe imaging at 750 nanometers, and that would work with 2P, et cetera. So again, great collaboration. No, I didn't do anything. This is a shout out to my colleagues. These are the best in class voltage indicators. Sorry, I'm running a little slow due to technical difficulties and talking too much. Wanted to give you a quick update on some other things. We've made a lot of neurotransmitter sensors. So if you're going to make one neurotransmitter sensor, it should obviously be for glutamate. So we did this. So we published the iGlu Sniffer 1.0, I believe, in 2015. Then this is the 2.0 version, where you can see you get really nice signal and noise. These are in, this is ferret visual cortex showing it moving gratings, and you can get these beautiful single spine orientation selectivity responses. So, but again, Janelia is always about trying to make them better and better. So Kasper Pigorsky and Abi Agarwal, whom I already mentioned for the SLAP microscopy, have done a fabulous job making a 3.0 version of iGlu Sniffer. So there are 2 flavors, 3A and 3B, with a bit of a trade-off in kinetics and signal to noise. So let's see. Okay, I guess that's the one video that maybe I should have tested it beforehand, but yeah, they're getting beautiful responses, and this is in cell culture. And oh, here we go. Video's playing now. Yeah, they just stress it a little bit with sucrose, and you get low level release events, and they have reason to believe that most of these are minis. So these are release of single vesicles of glutamate. So this is what they see. So again, you're getting all these little puncta, and you can see that you get mostly asynchronous release, and if you zoom in, they do seem fairly quantal. The size of the peaks is mostly uniform. So really think that this is going to be the future of high sensitivity glutamate imaging. And just to further back this up, in a publication from a few years ago, a collaboration with Dmitry Kuhlman and Dmitry Rusakov, we showed that by co-injection of a far red calcium indicator, you can indeed show that iGlu Sniffer has single vesicle sensitivity. And this was with the 2.0 version. So I really think that with the 3.0 version, that those are single vesicles they're picking up. If you're going to image a second neurotransmitter, it's obviously going to be GABA. So this is, again, great collaboration with the Dimitrys. They have a beautiful prep, a mouse model of focal epilepsy, where they inject a little pilocarpine, induces these seizing events, and these are the interictal spikes that you get between the seizing events. And you can see that there's a perfect match between the GABA, iGABA sniffer fluorescent signal and the electrical signal from an electrode in there. And then this is something we try to do with all our sensors, is we want to do controls to rule out artifacts. And so the blue and the red traces are responses to interictal spice with 2 good GABA sniffer variants. And then the black line, we have purposely mutated out GABA binding, but it's otherwise identical in targeting, photostability, pH sensitivity, et cetera. And there you get a flat line. So it really was GABA we were imaging with the sensor. And then again, always trying to make things better. So Genie screened a couple thousand variants of iGABA sniffer, and we get this, monumental increase in iGABA sniffer 2, which we're now, again, this is available. Just drop an email. And so you can see this is iGABA sniffer 1. It was not really picking up release events following single field stimuli, whereas now iGABA sniffer 2, you're really getting a pretty robust response. So that's impressive. We also have a negative going iGABA sniffer, and this is, we have these variants for a lot of things, but people are usually less interested in the ones that go down. You always want fluorescence to go up. But for GABA sniffer, this might be useful for disinhibition, to see a release of GABA, and then you would get a signal increase. And just to show you, I do think this is going to be useful. This is now high resolution imaging of a cultured neuron, and you can see that you're getting at these different fields of view along the cell body, dendrites, axon hillock. You're getting these really different release profiles for GABA. So I think that's going to be super useful. I am running short on time, so I will just summarize a lot of things for you. So we have a acetylcholine sniffer that works really well. So again, I mean, it works great in mouse and fish, but it might be most useful in fly, where acetylcholine is the prominent excitatory neurotransmitter instead of glutamate. We made a serotonin sensor in collaboration with Lin Tian, and this works really well in vivo. This is imaging from Viviana Gradinaru. Actually, it's not imaging. This is fibrophotometry. So the signals are good enough that you can just do photon counting, and you can see you get these really big changes during changes in sleep cycle with, for instance, this big change in basolateral amygdala from transition from REM to awake. We have an ATP sniffer. We have a glucose sniffer, and I'll just say, we made some reagents for optogenetics and Chem, and I will, I'll send you a recording to another talk where I managed to get to all that material. And again, I'll send you these slides and every slide I've ever given. So let me just wrap up. So all the GCamp 8s are available at Adgene. They're now shipping AAV 1 and 9. They hope to have AAV Retro ready to go by the end of June. For all the neurotransmitter sniffers, you can either email me or Casper for iGlu Sniffer 3, or really want to emphasize that if you email this address, geniereagents@genelia.hhmi.org, that will always work, even after I'm gone. So that's probably a good one to use. And then last slide, Genie is a huge collaboration, a lot of labs over a lot of time. And this wouldn't be possible without it. Thank you.
Thank you, Loren, for your time. I presume you have somewhere to run to, or maybe we can take.
Yeah, I can hang around.
Oh, okay.
So please ask questions, join, whatever. Send me emails for clarifications and things I didn't get to.
Okay, so just in case somebody's shy, I'm going to break the ice here. I know people are always trying to push you for faster time scales. I'm going to go the opposite direction. What about long-term imaging, problems with photo bleaching, and let's think about hours, maybe imaging for hours or even days.
Well, yeah, I mean, so the photo stability of the G-camps and the sniffers is astonishing. I think the reason is that once the delta F over F starts to get really high, like, 10, 20, 30, something like that, then your sensor is spending a lot of time in the dark state. And when it's dark, it can't be bleached. And so the photo stability of these things is really good compared to like a fret indicator. There you're getting, you get the signal comes from the change in distance and relative orientation, but both fluorophores are always bright. And so they're always bleachable. And so fret indicators bleach pretty quickly. So yeah, no, people have done, yeah, there are mice that have been imaged with GCamp for over 2 years. And it's sufficiently bright and photostable that it even looks good under 3-photon excitation, which, the photon fluxes there are truly staggering, but, still holds up well.
Nice, nice. We have a question here that says, do you think the red sensors will be able to perform as well as the green ones? You mentioned it's really hard. Can you explain why in more detail?
Yeah. So, I mean, I think so the first part of the question, no, I don't think the reds will ever be as good as the greens, maybe with a couple exceptions. And there's a couple reasons. So, as I mentioned with the red dyes, so to make something that's fluorescent in the red, the chromophore has to get bigger and bigger. And so for the fluorescent proteins, there are more and more catalytic steps that are required to make it. And so more things that can go wrong. So, when we made the GCamp8, we swapped in these like 60 peptides. For the green ones, I think that almost 50 of those worked, like with no optimization. I mean, they weren't perfect. They were all worse than GCamp6 to start, but they were all still green and most of them were still sensors. When we did that with the red sensors, so RCamp and RGecko, I think only like 3 of them were still sensors, and I think maybe 5 or 6 of them were red. The rest, it just broke, folding, maturation, etc. And there are other pathologies. So a lot of the red fluorophores bleed into the green. So, so this is another thing to watch out for, is that if you're using something like TD tomato as your red protein, if you switch over to GFP, it shows up pretty well in the GFP channel as well. And so you need to be careful with things like that. And then there are even more detailed pathologies that the reds have. Yeah, it's much harder.
Great. We have another one. This person is asking, I was wondering if deconvolution has been done with GCamp8F. to see how accurately it can detect spike trains.
Yeah, so I showed you guys a bit of the simultaneous eFIS with GCamp8. I don't think that that the rigorous analysis of like constructing receiver operator curves has been done to show like 95% detection at a less than 1% false positive rate, but that's in the works, and it's really good in terms of spike detection with low false positive rate.
We have another one. What would be a dream tool that you would like to have, but at the moment it's not possible, and whether a bigger community is needed, mixing people from different fields or, yeah.
Yeah. Oh, okay. Yeah, there's a couple. This is a great question. I love this question. Let's see. If I was dreaming, one thing I think we really need, and this is not just for neuroscience, I mean, I think any biological endeavor could use this, is a way to quickly erase a protein. And so you get like, I'll call it like a temporal knockout, ideally reversibly, because, we do these things, we make like a full CRISPR knockout or even like RNAi knockdown. These things are constitutive. And so the system has time to compensate. And so I would say a lot of the results from knockout animals, I mean, it's informative, but so many things have changed that it's super hard to interpret. Whereas if we had something where you can like shine a light and, within 1 second, there's no more signaling through NMDA receptors. And then you could turn the light off, and then a second later, there is signaling through NMDA receptors again. I think that would be pretty transformative. Then you could, conclusively say like the action of this protein at this super precise temporal interval in this exact cell is doing, this thing. There's progress, but I think that's like 5 or 10 years off. One that I think would be super useful, that I think is going to be a lot easier to make, this is something that we've been wrestling with lately, is a way to mark cells of interest for later reconstruction. There are a couple crude ways to do this. You could, you could look at CFOS. I mean, that's just too indirect. If you did like light-gated transcription, I think that's just way too slow and non-specific. We really want something that would be compatible with like antibody amplification and, the latest tricks like expansion microscopy, etc., and say like, hey, these 5 cells are doing something really cool when the animal is like navigating in a virtual maze. Let me go zap, 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 and then reconstruct those cells, light microscopy, electron microscopy, etc., and say, this is the ensemble of cells that were doing something super cool in behavior, and we then reconstructed them to, best possible resolution, figure out where they synapse, that sort of stuff. I think that'd be super, super powerful.
Yeah. I mean, looks like ending on a dream note. We have a lot of future ahead. With if we're dreaming that big, I think we have a lot of future ahead. I don't know if Miguel had another question.
I would just like to invite the people that join us. Maybe if they want to participate, of course, they're welcome to.
Absolutely, I've got time. So yeah, please, I'd love to meet you guys.
And anyone that wants to join and is assistant on YouTube, please join us.
Yeah.
Yes, we have a couple of participants already. If you're feeling comfortable, you can go ahead and unmute yourself if you want to ask questions. Some of you have already asked questions,
Hi, sorry, can I ask questions?
yes, please.
Please.
Hi, sorry. Hi from Denmark. So I was wondering if the GCAM that you guys are using, like GCAM 7B, that is for spine, do we know that if they affect the plasticity because they somehow buffer the calcium too? Has there any study on that one?
Yeah, I mean, yeah. So that's a great question, and you know, I'll even make the question a bit broader in terms of, you know, all these sensors and effectors that we're expressing. Yeah, how much is calcium buffering, glutamate buffering affecting intracellular signaling? These sorts of things keep me up at night. So I'll tell you what we know. So we, particularly for the ones that like the GCAMP 8s that are a lot faster, I don't think that that buffering is that much of a problem. I mean, so you asked a very specific question about the spines, and I can't really speak to that, but we did like, you know, so we patched cells that had been expressing GCAMP long-term. They had identical, you know, like orientation selectivity, membrane capacitance, resting membrane potential, etc. For, you know, so when cells get GCAMPed out and like turn into the zombies, I actually don't think that's from calcium buffering. I think that the calmodulin is messing up cell signaling within the cell. So there are a couple efforts to make these things more bio-orthogonal. You know, there's this thing called FGCAMP for fungal GCAMP that uses a fungal calmodulin and peptide that's supposed to be more bioorthogonal. Right now it has, you know, much worse SNR and kinetics, but we're trying to make it yeah, I need a little more time. But you know, we're trying to incorporate those mutations into, you know, so JGCAMP 9, et cetera. We'll have a couple more of those mutations. I can say a bit more for like glue sniffer. So for the sniffers, we don't think that buffering is a problem. There we were able to do much more precise measurements. So we had like neurons expressing glue sniffer, and then we would do 2-photon uncaging of glutamate at single spines and measure EPSPs at the soma. And those were identical. So, you know, we don't think, but I mean, there the rate constants are a lot faster. So the resonance time of the glutamate in the sensor is a lot slower. So it's a lot shorter. So I think it's the receptors are getting the normal complement of glutamate. But yeah, we should worry about these things. You know, there are papers that if you really express GCAMP at huge levels, it was like with the Tiger 2 with TET-TA amplification crossed with an EMX1 Cree mouse, which is a strong promoter. Then they got this epileptiform activity in the mouse, but we really think that's the exception. Mostly that the transgenic animals express at lower levels of sensor than AAV infection, and so I think they should be good for long-term imaging without messing up the mouse too much. but you're right, we need to be worried about these things.
All right. If anybody has any other question, I think this is a good moment to ask.
That's my last questions. So one experiment I'm thinking, I don't know how it will be effective or not. If I'm expressing, let's say, an RCAM in postsynaptic regions, and my presynaptic regions expresses some, let's say, OCHIF, some channel rhodopsin, and then I'm gonna look at activate those axons with the blue light. So usually I'm gonna use, like, I don't know, 15 milliwatt of light powers. How that would affect the activity of RCAM? Would it make some complicate my data, my reading?
So with RCamp, not so much. With RGecko, yes. So, you know, I mentioned that there were complications of the reds that were deeper into the weeds. So one of those with RGecko is that when you tickle it with blue light, you get these big responses. It's much lower in JRGecko 1A than it was in it's not lower, the signal and noise is higher, and so the contribution of that photoactivation is less comparatively. But yeah, no, that's something you need to worry about with RGecko. With RCamp, less so. That doesn't seem to photo switch.
Thank you.
All right. Thank you all for coming here. It was a blast. I mean, it's kind of drinking from the fire hose here. So we really appreciate you taking the time and explaining everything so thoroughly. And we will share the materials, of course. And
yeah, and everyone just email me, email Genie Reagents, so you can get all the text for the exact emails and whatever from the slide deck that will be available. So yeah, I hope to hear more from you guys with more questions and ideas for collaborations and new tools.
Awesome, awesome. And just to remind everyone, next week we'll have people from AdGene. So if you really want to know more about AdGene next week, we'll have them. So thank you for coming again, and see you soon.
That's amazing. Okay.